Every cell in the human body relies on protein metabolism to survive. Breaking down amino acids is how we build tissues, synthesize enzymes, and maintain basic cellular functions. But this continuous metabolic furnace produces a highly reactive byproduct. Cleaving an amino acid releases a nitrogen compound, generating ammonia, or NH4+. Ammonia is exceptionally volatile. If it escapes the local cellular environment and accumulates in the systemic bloodstream, it readily crosses the blood-brain barrier. There, it acts as a severe neurotoxin, rapidly degrading neural function and inducing coma. The body is therefore forced into a strict metabolic mandate. It must continuously neutralize and expel this stream of toxic waste, but it has to do so without consuming more energy than the organism produces to survive. This creates a logistical problem that no single organ or cell type can solve in isolation. You need chemical processing to disarm the toxin and physical plumbing to remove it from the body. Human survival relies entirely on a coupled two-stage pipeline, a complex biochemical conversion of the waste, followed immediately by brute force physical extraction. The first half of this pipeline is the liver. It acts as a massive biochemical reactor, capturing volatile ammonia and converting it into a safe, inert molecule called urea. The second half of the pipeline consists of the kidneys. They operate as a mechanical filtration plant, physically extracting that newly formed urea out of the bloodstream. Together, this integrated two-organ system forms the hepatorenal axis. Because the liver and kidneys are physically separated, both organs are entirely dependent on the vascular network and systemic blood pressure to bridge their functions. If the blood stops flowing at pressure, the pipeline breaks. To understand exactly how localized liver damage can eventually trigger total kidney failure, we first have to trace the precise molecular journey of nitrogen from cellular waste inside the liver to urinary excretion. We start inside a hepatic cell in the liver. Within this cell, there is a strict anatomical boundary separating the central mitochondrion from the surrounding fluid of the cytosol. This boundary houses the Krebs-Henselite, or urea cycle. This is the liver's specialized 5-step enzymatic detoxification engine. This diagram shows the cycle's rate-limiting first step, occurring exclusively inside the mitochondria. The enzyme carbamoyl phosphate synthase 1, or CPS1, condenses toxic ammonia with carbon dioxide. This initial reaction carries a steep metabolic cost, immediately consuming 2 ATP molecules. The liver burns this energy to securely lock the volatile ammonia into a highly stable molecular structure called carbamoyl phosphate, neutralizing the immediate threat. Next, that carbamoyl phosphate combines with an amino acid called ornithine to form a new molecule, citrulline. Once formed, citrulline crosses the mitochondrial membrane. It carries the trapped nitrogen waste out of the mitochondrion and into the wider cellular cytosol. The liver spends premium cellular energy up front to package a lethal neurotoxin into a form that is safe enough to transport. Out in the cytosol, the cycle incorporates a second amino group. Aspartate is added to the citrulline to form a larger complex known as arginosuccinate. This specific condensation reaction costs another ATP equivalent, further taxing the cell's baseline energy reserves. Now the molecule is ready to be broken apart. An enzyme cleaves the arginosuccinate, yielding 2 distinct molecules, arginine, which is the direct precursor to urea, and fumarate. This diagram illustrates a concept called bicyclic integration. Notice how the expelled fumarate flows rightward, creating a direct biochemical bridge from the urea cycle into the TCA, or Krebs cycle. Recycling fumarate into the TCA cycle allows the cell to recover immense metabolic value. Look at the energy totals. The TCA cycle generates 12 ATP, far offsetting the 4 ATP consumed to process the urea. Because of this integration, the high ATP yield of the TCA cycle ensures the overall nitrogen detoxification system runs on a net positive energy balance. In the final step, the enzyme arginase cleaves the arginine. This yields safe, inert urea and regenerates ornithine to seamlessly restart the cycle. The liver has successfully neutralized a fatal neurotoxin without bankrupting the cell's energy economy. The safe urea byproduct is then released into the blood. That newly synthesized urea molecule leaves the liver and enters the high-speed systemic bloodstream. The blood carries it directly to the kidneys, the body's dedicated mechanical filtration plants. Zooming down to the microscopic level reveals the nephron. A single kidney packs over 1 million of these individual functional units. At the head of each nephron sits the glomerulus, a high-pressure capillary tuft housed inside a cup-shaped structure called Bowman's capsule. The wall of this capillary forms the glomerular filtration barrier. This is the ultimate mechanical sieve for the human bloodstream. The barrier is composed of 3 precise layers. First, the blood encounters highly fenestrated endothelial cells, riddled with microscopic pores. Next, the fluid hits a dense basement membrane. Finally, it passes through specialized, interlocking podocyte foot processes that wrap around the exterior of the capillary. Unlike the liver's complex chemical reactions, the nephron performs a purely physical separation. It relies entirely on microscopic structural integrity to keep vital blood cells and proteins inside the vessel while letting waste out. Because this is a mechanical filter, the driving mechanism is passive. The kidney expends absolutely no local ATP to push the fluid through the barrier. Instead, filtration is driven exclusively by intracapillary hydrostatic pressure. This is simply the systemic blood pressure from the beating heart, pushing fluid forcefully against the glomerular sieve. Under this pressure, small molecules like water and urea are forced through the 3 layers. Large proteins and red blood cells bounce off and continue circulating. The filtrate that makes it through flows out of Bowman's space and travels down the long tubular loop of Henle. As the filtrate travels down the collecting ducts, water is reabsorbed into the body. This concentrates the remaining urea into urine, ready for excretion. This physical extraction process works perfectly under one condition. The systemic blood pressure must remain high enough to manually force fluid through the sieve. Pulling back to a systemic view of the vascular network, we examine the core vulnerability of the hepatorenal axis, its absolute reliance on cardiovascular hemodynamics. That vulnerability is exposed by hepatic cirrhosis, severe architectural scarring of the liver tissue. When the liver sustains severe structural damage, it triggers massive pathological fluid shifts and extreme vasodilation throughout the abdominal circulation. This sudden widening of the abdominal vessels acts like a hydraulic trapdoor, pooling massive amounts of blood in the gut, away from the central circulation. The direct consequence is a precipitous drop of effective systemic blood volume, causing arterial blood pressure to plummet. By dilating the abdominal vessels, the failing liver actively destroys the hydraulic pressure required by the rest of the body. This hemodynamic collapse leads to hepatorenal syndrome, a clinical paradox where structurally perfect kidneys acutely fail secondary to liver disease. Sensing the plummeting global blood pressure, the kidneys initiate a desperate survival response. They severely constrict their own local blood vessels in a flawed attempt to conserve volume for the rest of the body. Back inside the glomerulus, the fatal consequence of this extreme local vasoconstriction becomes clear. The intracapillary hydrostatic pressure drops to 0. This completely stalls the flow of blood into the glomerular tuft. Without that hydrostatic pressure pushing the fluid forward, the glomerular filtration rate drops to 0. The mechanical filter completely halts. Toxic nitrogenous waste, a condition clinically known as azotemia, rapidly accumulates in the stagnant bloodstream with nowhere to go. Biochemical mastery in the liver is useless without mechanical filtration in the kidney. And as hepatorenal syndrome proves, those mechanical filters are fatally dependent on the hemodynamic stability provided by the entire integrated system.